Well, I think it is 10 past the hour. So thank you everybody who's here. It's good to see you all online. So yeah, this is our final week 6 lecture in the beginners course. So we sort of have covered everything from sort of why is synthetic biology interesting and what is it, and then sort of the central dogma, how do we go from DNA to RNA to protein, and then how do I use synthetic biology to basically control that process and control what a cell does. And there's sort of so many different things you can do. And I think week 4, we really talked about like lots of different types of parts and sort of what's possible. And then last week, I gave an introduction to what this like practically looks like in the labs. Hopefully you have a mix of like the theory and also the practice. And we thought for this last lecture, we would do something a little bit different. So I'll be talking about cell biology by the numbers, which is basically a sort of a set of like useful values that you can sort of keep in your back pocket to help you think about biology. And we'll talk about that a bit more. And then we thought we would do something a little more advanced, but hopefully kind of inspiring to help you think about what kinds of things are possible with synthetic biology. You remember in week 1, we sort of asked people what they might be interested in, and something that came up was CRISPR-based systems, which I think has been mentioned before, but Ice is really going to kind of go into detail on like a certain type of application. So if you guys have any questions about that, and then I think he's going to talk a bit about some of his own research, which is actually quite cool and definitely at the advanced, multi-advanced level. So yeah, we will go ahead and dive in. So I'm going to, Ice, if you could keep an eye on the waiting room and let people in if they come in late.
Yeah, sure. I just like do the trick of like disable the waiting room so that like they come in right away.
Perfect. Okay. I will now. Okay. So here we are. All right, so everyone can see my screen, hopefully. Awesome. Okay, so yeah, let's jump in. So part 1, cell biology by the numbers. And basically, what this means is that there are some very specific numbers in cell biology that I think it is useful to know. And I want to talk a bit about why it's useful to know some of these numbers. So one is to like build a good mental model. So we've interacted with the idea of like, what is an E. coli cell, or what is a cerevisiae cell, or what is DNA, what is RNA, kind of as like words. But if you close your eyes, do you sort of have a picture of like what these things look like and what do they look like side by side? And I think having these good mental models helps you sort of process new information that you receive, and then also come up with your own ideas and be creative when you can sort of sit by yourself and think, okay, what kind of thing could I put in a bacterial cell? It might actually matter that you know how big a bacterial cell is to understand sort of what you could put inside it. Another important thing I think related to sort of learning new information is to get sanity checks. So you might hear us say something in a class, or maybe down the road, if you're doing research, you might be reading a published scientific paper in a journal that gives you some number. And it says like, we measured that these aggregates of cells were 100 microns across. And it's hard for you to know, is that number surprising or interesting? What does it mean that it's 100 microns? So again, maybe if you know the size of the cell or the type of cell they're talking about, you can know how many cells are present, and then maybe have a better idea of like what this thing looks like and if it's actually interesting. And then I think the last thing that knowing these numbers is useful for is to do a calculation, or like say you have some idea and you want to explore it with just pen and paper. Sometimes doing a little bit of thinking beforehand allows you to not have to do the experiment. And I know some of you are kind of new to synthetic biology, you're not working in a lab yet. You could think of these like back of the envelope calculations as a way to sort of explore some of your ideas. That's sort of why I think these numbers are useful. So again, we're kind of building our mental model and trying to have a way to think about new information we receive. Okay, so what we're going to talk about briefly are sizes of cells, which I mentioned, sizes of biomolecules, sizes of genomes, a particular biomolecule, a couple parameters related to time, and then I'm actually going to show you a website that's entirely dedicated to sort of organizing and getting some of these numbers, because there are a lot of researchers who are excited about collecting these things and making them available. So okay, to start with this, and feel free to unmute or put this in the chat. I thought it might be fun to have this be a little more interactive. So we've talked about 3 cell, like we call them chassis, or like model systems: bacteria, yeast, and mammalian cells. And I'm just curious, if you had to rank those by order of size, which one is the biggest and which one is the smallest? You can take a moment to think about it, or you can also type something in the chat if you think you have a good guess. Okay, so maybe you have some idea in your mind about which one of these is the biggest, but it actually goes like this. So bacteria are the smallest. They're about 1 micron across. Yeast are sort of in the middle at 3 microns. And then mammalian cells, so these are cell lines we've talked about, like HeLa cells. Other cells from other organisms like CHO or mouse cell lines are also in this category. They're about 10 microns across. So these numbers are useful, I think, in 2 ways. So one thing we can notice is the relative difference between bacteria and mammalian cells. So mammalian cells are 10 times bigger sort of across. And remember that this also means they are potentially up to 1,000 times bigger in terms of volume, because you take the radius and you cube it. So if you want to compare the biology of a bacterial cell and a mammalian cell, in your brain, you can think a mammalian cell is quite large. These are also valuable in an absolute sense, though, because you can think about, okay, a micron is 1/1000th of a millimeter. So I think a millimeter, I can see something that's a millimeter across, but something that's 1/1000th of a millimeter, much harder to see, which, okay, we're familiar with not being able to see cells. But you can also think about the fact that a human hair, these can range from 10 to 100 microns. So probably a like reasonably large mammalian cell is close to like the radius of one of your hairs, which is quite small. Okay, so this I think is a really useful number. Here also are some nice pictures from a textbook that's actually called Cell Biology by the Numbers, also to give you a sense of these relative sizes. And a lot of people, there's actually a great artist, I'll maybe have to send his name later, but he tries to draw pictures to also help people build mental models. So for here's an example where we have an E. coli cell compared to a yeast cell. So you can already see the fact that they're like 3 to 5 times bigger. And you also see that there's more stuff in the yeast cell. Remember, we talked about E. coli are examples of prokaryotes, so they don't have a nucleus, whereas yeast cells are eukaryotes. So they not only have the nucleus, which is this brown circle, but they also have organelles. So we see mitochondria, we see some vacuoles, we see endoplasmic reticulum. So you can just kind of keep this in your mind. This looks like a busy, bustling city, whereas this one's sort of a little simpler. Something else I also like to see sort of interesting is this flagellum. You may be familiar with the idea that bacteria swim and have stuff sticking out of their cell surface. This thing is actually as big as the cell itself, which again might be surprising if you're trying to think about, okay, how big is its like tail, basically. This book also has a nice picture of a mammalian cell. So again, the E. coli and yeast would be much, much smaller relative to this picture. We see similar types of organelles, but you see there's just more mitochondria. The shape is different. And actually it doesn't have, you can just hear there's a yellow border around it, and this is the cell wall, which we talked about. This one doesn't have a cell wall, so that also affects its shape. So maybe in the picture you have of a mammalian cell in your brain, you can imagine it being more irregularly shaped. Okay, wait, did I go? Oh, then I wanted to talk a bit about sizes of biomolecules. You don't have to memorize these, but I thought something was interesting, as you can think of the nucleus as being about 10% of the volume of the cell. The cell membrane itself is 4 to 10 nanometers. So we could compare that to, say, our, I guess, 1 micron cell. That would be about 0.1%. So the membrane really is this like thin skin on the outside. Our average protein diameter is 3 to 6 nanometers. We know about the size of a water molecule is 0.3 nanometers. But maybe the main thing to take away from this is you could do some kind of calculation about what's the maximum number of proteins or water molecules you can put inside a cell. It's sort of, I think, counterintuitive to think that there's like a limit on the number of these things, but we know because there's a volumetric constraint. You can't pack kind of an infinite number of things in there. Okay, here's my next sort of thought experiment question. If you had to rank these in order of size, which do you think is the biggest and which is the smallest? So when we talked about the central dogma, you have DNA that encodes a gene, you have mRNA that encodes the gene, and then you have the protein that's encoded by that gene. So I guess maybe which of these do you think is the biggest? Feel free to put a guess in the chat, but just think about which one you think might be the biggest. Okay, protein, yeah. Okay, we have another guess for protein. Oh man, everyone's guessing protein. Okay, good. This is the intuitive guess. And you know why? It's because when we draw pictures of DNA, we just draw this like little string. And then when we draw a protein, we draw this like big circle blob. So in your brain, the protein is bigger than the DNA and the mRNA. But actually, this is kind of a scale drawing comparing the size of an mRNA molecule to a protein. So actually the protein, of those three things on the list, the protein is actually the smallest. And we'll talk through a couple reasons why, but this picture shows some of those things. So first of all, this long chain molecule is sort of extended and not tightly folded. That's like one thing. But we also see that there's kind of more stuff here. We didn't talk about this too much in the course, but when you have your mRNA molecule, it includes things like these untranslated regions or a poly A tail to make the mRNA molecule more stable. There's actually additional stuff on the mRNA molecule that's unrelated to the final amino acid sequence. And here are a couple more things that you could keep in mind. So for example, DNA actually is 2 strands together rather than 1 strand. So we'll use that to argue that the DNA is a little bit bigger. It might also include, in a eukaryotic case where there's some mRNA processing, both exons and introns. Remember, the introns are sort of spliced out of the mRNA. The RNA, like we saw in that picture, can include untranslated regions. I forgot to type it here, but the other thing we can remember is that for every 1 amino acid, you need 3 nucleotides. So even if those things were the same size, you should expect the mRNA to just have more atoms in it because there are, again, 3 nucleotides per amino acid. But we can also, going back to these pictures from lecture 1, compare the sizes of the sort of basic nucleotide versus the basic amino acid. And you see the nucleotide has a lot of pieces on it. It's not just this base, but we have a sugar and we have a phosphate, whereas the amino acid just has this smaller carboxyl group and amino group. I think it's hard to remember these things because we sometimes think about them abstractly. Like you can just summarize a nucleotide or amino acid with a single letter. But sometimes it's helpful to remember these things are composed of atoms, and we can do things like count the number of atoms to compare their sizes. It's a little bit unusual, but it can be helpful sometimes. Okay, maybe last question, I think, is which organisms do you think have the largest genomes? Well, we can do our regular E. coli, yeast, human cells again. Yeah, feel free to put in the chat like bacteria, yeast, humans. I'll give you a hint. This is, oh, ice float's plant.
They're big.
Okay. Yeah. Ice cheated a little bit, or ice knows, but this is great. Okay, so thank you so much, everyone, for putting things. Okay, this one was less of a trick question. So here is a chart from Wikipedia showing the ranges of sizes. So we see our bacteria are sort of down here. We have our fungi and molds that are here. And then we have mammals, which includes humans, up here. But you see which one, so everyone who answered mammalian cells, yes, that's correct. We sort of roughly, it goes with size. But there's this interesting thing that actually, if you ask which organism has the largest genome, it's actually plants. And there's sort of a fun reason for that, which is that many plants have duplicated their genomes as many as 8 times. In fact, if you've done a DNA extraction protocol, people love to use strawberries for this because they just have so much DNA because they've duplicated their genomes. And there's at least one reason for this, is that genome duplication actually helps if you have a larger cell. And leaf cells tend to be quite big. You might even be able to sort of like look at them under a microscope and see that they're larger, so that helps. And sort of the intuition for that is like the more DNA you have, you can sort of get more protein because you basically have more instructions. Okay, and okay, so here are just some useful sizes. We sort of saw this on the previous thing, but in terms of absolute numbers, you have about 5 times 10 to the 6 base pairs in an E. coli genome. It's about twice as big for yeast. And then for humans, our genomes are about 1,000 times bigger than E. coli. which is pretty cool. And you can think that that roughly scales with the volume. Remember, we like roughly calculated that in terms of volume, a human cell should be about 1,000 times bigger than a bacterial cell. Okay, I'll go a little bit more quickly through this last bit, but here are just some fun questions for you to sort of ponder, which are, how many genes do you think a cell needs? Like the smallest, simplest cell you can imagine. Should it have 10? Should it have 100, 1,000, even more? And maybe how many more genes would you need to become more complex? Like how many more genes does a human need than a yeast gene? Okay, and sorry, I should clarify a little bit. This is different from the previous question because you remember that the genome is like the full DNA molecule that's inside. Not all of that encodes for proteins. The genes are just the parts of the genome that are encoding the proteins. So this is actually slightly a different question. And remember, we saw on the previous slide that the human genome is about 1,000 times bigger than the bacterial genome. But if we actually look at the number of genes, so an E. coli cell has about 4,000 genes. Cerevisiae has about 6,000 genes. And C. elegans, mice, and humans have about 20,000 genes. So I think this is a little bit surprising because this shows us that a larger genome doesn't equal more genes, or proportionally more genes, let's say. Because we can think about the fact, again, we have our DNA is about 1,000 times longer, but we have about 5 times as many genes. And part of the reason for that is one way to make your genome more complex is not to actually encode more proteins, but to put more regulatory DNA. Basically give yourself sort of more control over when genes are expressed, sort of, and in what locations. Okay, so a couple time parameters. We talked a bit about how synthetic biology is faster in bacteria and yeast than in humans. This is part of the reason because an E. coli can basically replicate itself in 20 to 40 minutes, but for a human cell, it will take about a day for it to double. So this just kind of slows everything down. But again, this maybe matches our intuition that the human cell is bigger. To like make all the proteins, all the molecules you need to make a whole new cell, it might take a little longer. I thought this was also interesting, again, thinking about how long does it take a protein to diffuse across a cell? Like how long do you have to wait for two things to find each other? This takes 1,000 times longer in a human cell than in an E. coli cell. And also something that we can think about as well is how long would it take to translate a protein, like basically a ribosome to move from one side of the mRNA molecule to the other. And we might think of this as happening like super, super fast, but actually it takes about 10 seconds, which is like a time you can wait for. So say there's a ribosome in your body right now. If you wait 10 seconds, that's enough time for it to make an 100 amino acid protein. Okay, really quickly then, I want to introduce you to this website called BioNumbers. The link here, if it works, yeah, actually takes you to a PDF, which includes a lot of the numbers that I mentioned, plus some additional ones. But if you go to the home area, there's this like search bar where you can just type things in. So something I think, let's see, what would be interesting. So we talked about, I'm just gonna actually put in rhodopsin because that's a protein we talked about in one of the homeworks. And you can see kind of all the numbers it has. So somebody's calculated, let's see, the catalytic rate, I guess maybe like how quickly it can sort of do this reaction, I think with like incident photons. They've also calculated how quickly it is regenerated. Oh, here's one I was curious about. Yeah, how many rhodopsin molecules do you need on a rod cell? So this is one of the cell types in the eye that expresses rhodopsin, 40 million, which sounds like a lot, but you guys now have the sort of numbers that you could calculate maybe how many, I guess, proteins do you expect there to be sort of like per unit area on the cell surface? Like how dense are they? Yeah, but there's many, many numbers here related to lots of different things. So if you're curious about something, you can go check it out there. Yeah, sorry, that was a little bit fast, but hopefully that gave you some useful things, and maybe there are some surprising things in there. Like everybody always thinks that the proteins are the biggest, but it's actually the mRNA and the DNA that are larger. So hopefully that was useful. Does anybody have any questions? Otherwise, I might hand it off to Ice next, but just wanted to see if anybody wanted to chat. Oh yeah, we'll put that link. I'll put the link in the chat right now, and it'll be in the slides, and we'll also that'll be in the email. Okay, awesome. Well, thank you guys for interacting. I'm gonna hand it off to Ice then.
Thanks a lot, Heidi. So like one thing I would like to say on top of those by own numbers, like content is that like if you have any question or like you want to start some conversation about these numbers, please feel free to do it in Discord as well, because like some of these numbers will turn to be pretty interesting for everyone, I would say, because it's something that like you can imagine it in another way, because like most of the time it could be just me, but like when I start learning biology, people sort of like try to split between like people who like biology, people who like math, and like maybe like we don't get along, but actually like we do need a lot of numbers and math in order to like understand biology better. And you see like something in the field called like quantitative biology that came up in the synthetic biology a lot more these days, because there are a lot of data that we've seen, and we need more models and like methods in order to understand this thing better. So these numbers or this website is something very important that will help us explore the research in the field much further. So we'll change gear a bit into like introduce you about like something that was the tools that has been used in the field for a while, and something that sort of like I would say that is revolutionized the field to a certain extent, because like after the emergence of CRISPR technologies, the ability to edit and regulate genes has changed completely, especially in the terms of like how much it would cost to edit a single gene, for example. So this is the course material that I used to like teach it in a longer time period, but like I would like summarize it up. So if you have any question during this lecture, just feel free to put into the chat, or like you can unmute yourself and like interrupt me as well if you have any question, because like some part may be a little bit there might be a lot of parts in how to make the CRISPR stuff work. So it's better to understand what's going on in slide number 2 before like we go into slide number 5, that you have like some dependencies on the previous slide, something like that. Okay, so again, the title for today's talk is going to be about CRISPR technologies, and we'll talk about gene editing or gene regulations. I'll focus on gene editing first, because it's the one that has received the recent Nobel Prize in 2020, and like we are using the materials that actually like provided by the IGI, or Innovative Genomics Institute at UC Berkeley, where Jennifer Doudna, one of the Nobel laureate, has been part of, and they have sponsored kind of like these teaching materials and like some Wikipedia like or encyclopedia like system that you can go and take a look. So the link is right here. I'll put the link at the end, like to the chat at the end of the lecture as well. But you see a lot of materials that came from this website. So just to give it an overview of like what is the difference between gene editing and gene regulations, as Heidi said in the last few slides, that like even though the DNA in the organism keep growing and growing, it doesn't mean that you have more genes. You may have like more regulatory genes or like regulatory parts into the system. So that's why like we are trying to not just edit the genes, but we try to regulate the genes as well. But for the first part, we'll go and dive deeper into like how to do CRISPR-based gene editing first. In order to do that, I would like to like emphasize, our previous lecture again, that like we need to keep in mind that like the central dogma in molecular biology start from DNA and then go to RNA and protein. And then like if you do the edit at the DNA level, most of the time you can you can see the effects at the RNA and protein level as well. for CRISPR, it's kind of interesting because like the CRISPR tools comprise of like different parts, and it's actually in, encompass like all DNA, RNA, and protein. We'll get there in a second. But first question is sort of like, before CRISPR become a tool, what is it? So this is the 6 examples of method on how bacteria fight against virus. So keep in mind that like, even though we are talking about virus, it's not that dangerous to us if we were to work with bacterial virus, because virus is very specific, and they usually have a very specific host in that they will want to invade. So in this case, is the different system that the bacteria has installed the tools in order to fight with the virus that might be coming to invade them. There are several methods, like when the virus is coming to invade the bacterial cells, you can try to like put something on the surface and kick it out, put something in in between the surface of bacteria and the inside, so that like it block the virus from attacking properly. When the virus come into the cell, you can have some machinery that get rid of those genetic materials that come in, either like block it from being read or cut it out, or maybe like you can like program the cell so that the cell that was attacked is sort of like just died out, so that it doesn't survive and like keep being the resources for the virus. But the one that we are interested in today is the CRISPR system, that stands for clustered regularly interspaced short palindromic sequences. Repeats. So CRISPR is sort of like something that was very, very specific. And the method of like doing the CRISPR is sort of like the bacteria would just like collect the information of virus so that it remember what the virus is, or what the virus genetic material is, so that they can fight with the virus properly and specifically. So this is the overview of the process. If we were to divide it into 3 key functions, it would be first trying to record what comes in. So like if there is a foreign DNA from bacteriophage or the bacterial virus, the system of CRISPR will take these part of DNA and put it into its own genome. So it record that like these DNA or these like genetic materials are something harmful. And if it ever sees it again, let's just get rid of that. So that's the first one. And the second one is the biogenesis of the machineries. So after we collect it as a DNA, it's similar to the transcription process, that like these DNA will be transcribed into some RNA, and this is called the CRISPR array, and it will be transcribed into this kind of long RNA molecule. With this long RNA molecule, there was some processing system that can split them into the short sequence, and this short sequence will combine with the Cas protein. So it's a complex of RNA and protein, so that it becomes something that was active, like this complex, that can bind and target the foreign DNA or RNA, depending on the system of CRISPR. And then the idea is like, if you find these foreign DNA, let's just kick it out or edit it. So that's the that's the overview of how CRISPR was being in action in the nature. So wait, I see some question in the chat. Okay, it's from the previous one. I'll move forward. Okay, so the question that we have seen, like when we are trying to target these sort of DNA, is that like, if you're targeting these DNA of the foreign organism, how can you not target yourself and keep cutting your own genome? The nature came up with like a very cool like strategy, where it has like another like requirements in order to do the CRISPR targeting, to have the PAM sequence. So the PAM stand for the protospacer adjacent motifs. So these sort of sequence, we call them protospacer. So in order to target something right here, you need a PAM as well. So if your DNA in the bacterial cells doesn't have PAM, then it will not be cut. but only if the foreign DNA from virus right here has this sort of PAM sequence, then it will be cut. That's why, like, you can keep these DNA in the cells as long as possible, and it will just stay stable without being cut. So so that means, like, whenever these CRISPR system is going to be enact, it will have to like find these target DNA that has one, the PAM sequences, and two, the identical sequence of the complementary sequence of DNA and RNA in order to do the cutting or the binding. And that's it. That's why, the CRISPR is very powerful, because it can be programmed to a certain extent. You need the PAM, and you also like need the complementary DNA and RNA, so that, like, you can like program it very easily. And one thing to mention in terms of number is that, like, these RNA are relatively short. Like, it could be only 20 bases in the RNA sequence that was unique in order to target these things. And one trip and takes and tricks here is that, like, these short DNA or RNA can be synthesized in a relatively cheap manner right now. That's why, like, CRISPR has become that's why CRISPR has become sort of like a revolution tools that people can use, because, like, it's not that expensive to synthesize these small DNA or RNA compared to the big protein. Okay, so even though, like, we go through the overview of what CRISPR system is, there are actually different CRISPR systems in, the nature and how we repurpose them as well. So, for example, they do have PAM requirements, but if you take different CRISPR machines, like CRISPR protein from different organisms, they may have different PAM requirements. And with these PAM requirements, they can also, like, have different property of either targeting the DNA or targeting the RNA. You can think about, like, if you stop the invading, like, virus that was more of the RNA virus, then targeting the RNA might be better as well. And there was also, like, the components of the guide RNA and the components of the CRISPR itself, that, like, it could be just a single protein, it could be a multiple proteins coming together, or the guide RNA that was used for this, like, locating the actual DNA could be just a single part, or it could be multi-part that has to come together. And at the end of the day, it could make, like, a different kind of double strand break. It can make, like, a clean cut, which is a blunt end, or it can make, like, a sticky end. Like, if you recall, like, our previous lecture about the cloning, then the sticky end and the blunt end could make it a little bit different in terms of genome editing. And at the end, it can be actuated by the presence of the guide, and like, it can just keep cutting with nonstop and turn the enzyme to be active, something like that. So if the enzyme was activated by this RNA, we can repurpose that for some other applications. For example, like, use this CRISPR system for detection of the foreign DNA or RNA, and that's how we make the CRISPR diagnostic tools during the COVID time. Okay. So that's sort of like the overview of the CRISPR system. If you were to dig deeper into, like, how the CRISPR work, there's a lot more, but that's an overview. So, like, let's take a step further into, like, how do we make it into the genome editing. So right here in this figure, we show you, like, how the immune system of the CRISPR work in the bacterial cells. Let's think about, like, if we were to make it work in another system where, like, we want it for the genome editing instead, what can we do? So maybe, like, the key challenges in order to, like, repurpose the two to the new system is maybe, like, how do we make sure that we can have these complexes, and how do we optimize them to make sure that, like, they can do this kind of cutting, or, like, whatever downstream editing that we want to do. So this figure is a little bit complicated, but, like, we'll dig deeper into each step of these so that we get a clearer understanding of what the CRISPR is. So this is the a simple separation of, like, this core CRISPR complex. So you have this core CRISPR complex where what we call the complex of, like, ribonucleoprotein, where these this protein of the Cas protein and the RNA form the complex together and then target the DNA. So if we separate them into 3 different parts, into DNA, RNA, and protein, you see that like this Cas protein and the guanine and DNA complex were sort of like the 3 machineries from the central dogma. And you can develop like the tools based on each own system. And for each of these system, we can like further optimize it further to make sure that like you can make the tools that will be better. Okay, I can see some question in the chat saying I'm not understanding what CRISPR is. Let's take let's keep moving for this thing, and like if you have specific question, I'll like I'll go further, and maybe like I'll keep reiterate what the CRISPR is throughout the lecture, and maybe like that will help you a bit. But if you still like a little bit obscure, that's still okay, because this system is not that easy. So we will go for like it's a simple way of like how do we call CRISPR, or like the difficult and an in-depth system that we're still trying to understand it right now in the biology. So it's also very new, so it's not something that people know a lot, honestly, and it's still getting rediscovered and relearn and unlearn all the time for this system. So like if you don't get it, that's completely okay. The other question is like, how does siRNA work, and is it present in nature? siRNA might be another topic, and I would say I would skip that for now and like talk about CRISPR first for the time that we have. And the last question is CRISPR part of DNA? CRISPR is a system, and for CRISPR to be active, it needs all these 3 machineries. Like you need the protein, and you need the RNA to form the complex and then target the DNA. So was encoded from the DNA, but it is not actually part of DNA itself, if that makes sense. Okay, when we have this part, let's move forward. So if you were to engineer this system to be better, you can engineer at each components individually. And this is like what it actually do in the actual system. So like, if you see this complex, it can find the target DNA, it can cause the double strand break. This is something that we want to do most of the time in the genome engineering. Like if you break the DNA, then the bacterial cells or like the mammalian cells, whatever system, will try to fix it. And by fixing it, it can lead to some system called like non-homologous end joining, but where like they fix it, but they fix it in a way that was not completely the same as what it used to be, and you can disrupt the gene. Or you can do homology directed repair by supplying another DNA into the system, so that you can replace the system, like the repair system, the DNA repair system will recognize this thing and replace this part with the new DNA, so that you can make a new edits to this stuff. So how do we make a metaphor, like compare to some sort of system? Let's say we have a bicycle here, and when you ride a bicycle, then you have to brake sometimes, right? But if you disrupt this gene or like this function of the bicycle by get rid of the brakes, then your bicycle cannot brake anymore, and that's something that was completely changed in the system. Or if you're trying to like do a little bit different, but like modify the brake, let's say that the regular bicycle that we have right here is braking at the rear wheel. If you're trying to edit it, like by taking the in this case, like the example was like take the gene for leaf color and replace that with the gene of flower color, then the color of the flower will change color. In that case, then like you sort of like making a little bit of change to the brakes so that it brake in the front wheel instead. So like the function may still remain, but it's not functioning as the same as the previous one. So these are like the overview of gene editing. Like honestly, when you go deeper to like how to edit the genes, there'll be a lot more complications, because we have to care about the promoter, the gene, and the terminator and everything, right? So this is sort of a simplification, but like the long story short, the takeaway for this one is that like you can cause double strand break, and you can make an edit of where the double strand break occur. So what can we do? When you do this double strand break, you can do an edit on not just the bacterial genome, but also the human genome. And these CRISPR editing tools has been applied to cure some genetic disease. For example, these defective pancreatic cells or defective red blood cells, if we fix some of these genes, what you can find is sort of like you can turn into the healthy cells, and that genetic disease that used to exist in particular individuals can be fixed or partly fixed, and that's a big thing. And the CRISPR gene therapy tools has recently been like approved from the regulatory part. So like in the next few years, we may see like expansion or like more development on these like curing of genetic disease coming up. So keep an eye on, and it has like a very a very interesting like development in the past 10 years. So everyone is all excited with this like using the CRISPR editing for curing disease. What if like we want to do it in a different way? Let's say that we have some limited amount of food and we want to make more food. What can we do, and what can we use CRISPR for? So since CRISPR can edit gene, right? CRISPR gene editing can be used for like editing like any particular DNA. We can sort of like edit some genes in in the plant or in the the feedstock or bacteria that we've been using, so that like it can improve the productivity of what we are trying to make. And that means like maybe we can make more food with this system. So these are an overview of what is going on. I'm not going to go into detail, but the long story short is that like you can make new functions to the food or like the system that you're trying to make. Maybe you can make food, you can make plant grow faster, or you can make plant give more fruits, or like higher quality seeds, so that we can combat the food insecurity, or like getting higher amount of food for the world, so that we thrive throughout these severe decades. So that's an overview of gene editing, and I hope like you can see that like what the simple homology directed repair can be doing. One thing I want to mention just briefly is that like we can actually edit the CRISPR system to combine with the other system that was available in the field, so that we can make like new novel tools. So I'm gonna give just two examples, which is like if we combine this CRISPR tool with the base editing technology, or chemically you can sort of like change one amino acid into another amino acid using the enzymes that has been used in the nature to edit this thing originally. So instead of like doing the cut, you can sort of like get there and edit the specific DNA without cutting at all. And why do we want to do this? It's because like if we want to disrupt the gene, maybe like you can't really predict what is the outcome. Or if you want to do like the homology directed repair, sometimes it's sort of like being pretty severe to the cells, and the efficiency might be not that high, because like thinking about you keep cutting the DNA, then the cells may not be that healthy. That's why if you just edit, like do a single edit to the target gene, maybe that was like a lot more healthy, and it is actually. There are some other applications these days, both on the health science and on the food side, to use the base editing to like do something similar to what we've done with the gene editing, like based on the cutting. And another thing that I would like to mention is that like we can do both like C to T and also A to G, and that means like we can do any edits, right? Because if we edit like on A on one end, then it will be G on the other end. So instead, if we edit A on the other strand, then we can sort of like doing the reverse editing as well. So that's sort of like the overview. One thing that I would like to emphasize right here is that like these specific enzymes that we're working on not that specific yet. So like if you're trying to target some Z to T editing right here, it may edit multiple C into multiple T, and that's something that we still working on to develop these further. The other thing that I would like to mention is the post-graining angle of the CRISPR. As I said, that like the guanine could be pretty cheap to order or to synthesize, so we can sort of like synthesize them as a pool and then just put them into the cells and if each cells get different guide RNAs, then they will be modified differently. So if we construct a big library and just do the whole screening, you can say that, like, when you put into the cells and we use the growth as a screening, you can try to see what is the edit that will be working properly and get the trait that you would like, either you want something like that. So like, let's see that we have like the original cells and we modify them to be like different color right here. If this purple cell is the only cell that survive, then you see which edit makes sense. This is very functional in terms of like trying to study what is the essential genes. For example, if you design like different guidances to target every genes in the genome, and you want to know like what is the important gene and what is not important. Because if it's not important, if you disrupt them, they can still grow properly. But if it's essential, then that cell will just die, right? Because it doesn't have the essential gene. So that's sort of like something that instead of like trying to go one by one of the editing, you can like throw everything into the same like one reaction, like one tube, and then see what is the outcome, something like that. So with another technology in the DNA sequencing, we can also go very deep in this kind of technology. So we may not have time to go specifically on like how we get this to be completed, but I believe, like, I hope you get the overview of like we can synthesize these like small RNA like in a relatively cheap manner and acquire the data that may, let's say if there's a $1 million question that we can solve with maybe a few $100, that sounds perfect, right? Okay. These are selected references, especially CRISPRpedia that was provided IGI. If you want to see further, how CRISPR work, please visit these websites or like these reviews that were available. So let's see if there is any outstanding question in the chat that I can address real quick. Otherwise, I'll go to the gene regulation part, which is my personal research project. And I will go through it briefly to see that, like, how it's like to do research in the field of synthetic biology, and how do we frame the question, and how do we achieve it, and what kind of like tricks that we've been using in the field so far, at least my like very specific field. Okay, I see question from Tito. How does CRISPR have ability to detect and edit specific gene of interest? And thanks, Heidi, for like responding in the chat. But yeah, one thing that I forgot to mention or like specify it is that like we use these guide RNA, and this RNA is actually doing the guiding job. So it will just try to locate the DNA that has a complementary sequence between, the RNA and DNA, so that you can target the gene very specifically. And how can we detect them? We can also do that, and that's how the CRISPR detection sort of work. Like if you detect anything, it may trigger some events. For example, if you use the CRISPR system that will like turn the CRISPR the Cas compound to be an active protein and start like cutting different DNA, then you can amplify the signal by turning the enzyme from inactive form into the active form. And if you have like, let's say, the COVID DNA, then the whole reaction may turn green, something like that. And another question is, are there any research based on CRISPR to help with AMR issue? AMR, I assume that was antimicrobial resistance. And the quick answer is yes, but it might be a little bit complicated because like it was something that was used in bacteria originally, and if you're trying to like use this system to solve something in the bacteria, we have to like design it appropriately, otherwise the system may just sort of like crash with each other. And I may not have enough time to get into that, but like I'll be happy to talk about it in Discord, or like you can email us sometime, and we can like provide like some further information.
I just wanted to add really quickly that the first research project I actually worked on in undergrad was an attempt to program CRISPR to target the bacteria's own genome as like an alternative to antibiotics. And I think there's some particular reasons about like why it's good or not, but yeah, we can definitely try that.
Yeah. It is like very likely that it can be possible, but there are some concerns in consideration that you don't just keep cutting your own DNA and like the cell just like dies off, something like that. Yeah, but it's a very cool research in the field. So so for my research in throughout my PhD is actually like doing the gene regulation angle of the CRISPR tools instead. So when we go deeper into the CRISPR system, in order to cut anything, it has to maintain the ability to bind the DNA first, and then has the nuclease activity, like has the scissor property to cut the specific part, right? So what if we change it a bit into something that can bind but doesn't cut? Then that will behave like, let's say you're on the road and there was some car that was like parking in the middle of the street, then you cannot move forward anymore. That's something pretty simple to like what we call CRISPR interference. You put the CRISPR complex in between or like close to where the gene is supposed to be read. In that case, you cannot read that gene anymore, but you didn't actually destroy that gene. So for the metaphor of like, you know, the street, you can sort of like, you can put the big truck in front on the street, and there'll be traffic, right? So you cannot read the genes anymore. But in any case, if something clear that truck out, then you know this gene can be read again, or the street can be function again. Compared to the gene editing, where you sort of like destroy the road and you cannot read it anymore. So this is something that we that I really like, mainly because like we can do some sort of like what we call dynamic programming, like it was reversible. So if we get rid of these like DCAS9 DNA binding DCAS9 system, we can like get the function back. The other way around is that like instead of like disrupting the gene, we can try to use this complex to activate the gene. Let's say there's some gene that was like weakly expressed and doesn't give a lot of function. So we can like put this CRISPR and also another effector that was, you know, that was used in the native system to activate the gene, put onto like somewhere that it's supposed to be, without relying on the DNA binding property of this activator. What we can have is like we can have like a programmable activation system that we can activate arbitrary genes. And with these two tools together, we can sort of like study, you know, what the effect is going to be if you activate one gene or repress one gene, or what is it going to be if you like, you know, apply this regulatory system to multiple genes at the same time. So that's sort of like when I come to the PhD program, I realized that like these tools Lays developed in bacteria, and that's what we are trying to do in my lab at University of Washington. So again, CRISPR-I is based on the collision model. This is a quick you know, like readout of like how the bacterial transcription start. Let's say like there's a long DNA right here. This is the graph that tells us like where the transcription sort of start. And if you apply CRISPR-I, what you can see is that like, well, that it cannot start anywhere after where the DCAS9 was bound to. So that means like this is based on like, you know, like a roadblock kind of idea. If you put like the CRISPR complex right here, you cannot read anywhere downstream anymore, something like that. So if you were to do activation, you have to put some effector in. But the way we do it is that we are trying to re-engineer this system a little bit further by like not engineering at the protein level, but engineer at the RNA level. So what has been done previously by my team is that like this RNA was modified so that like the RNA can recruit another complex, and then this complex can be reprogrammed so that it can perform whatever function we want it to be. And in this case, it could be the function of like activating the genes or repress the gene. And this complex is working pretty well in yeast, and we want to make it work as efficient as possible in bacteria. And that's what, you know, I'm trying to do right now. So the first thing that we have to do is that like we have to find what the activator would be in the bacteria so that like it can keep working functionally. We go through like, you know, several candidates in bacteria, and we find that like, you know, this and this protein called success is the most functional one in bacteria, and we can use both CRISPR-A and CRISPR-I together in the same system in order to like turn the gene to be, like from red cells to be like green cells, for example, like repress the race protein and activate the green protein. This is sort of like something similar to the traffic light. And if you are trying to make the bacteria like communicate between each other, like, hey, this is green light, let's do this, or like, hey, this is red light, stop doing this, something like that. We can sort of like use this simple programming to control how the bacterial cells or the bacterial community, like multiple cells, to work together as well. So even though this is a simple, multi-gene control figure, it can mean a lot in terms of like, how do we program biology? Okay, then since we have these tools available, if you were to engineer it to be like a much better tools, what can we do? And this is sort of like, what I've been doing in the past, 4 to 5 years, is trying to like get deeper into each part of this CRISPR complex, either like at the protein level, at the RNA level, or at the DNA level. And then we try to like, edit each part to make sure that it's the most effective. So let's start by like the DNA part. We cannot edit the endogenous DNA ourselves because that's relatively difficult, right? So what we do instead is we're trying to like make a synthetic gene and study what is the rules to target, or like what is the rules for like doing the appropriate CRISPR-A. One interesting that we found in this like CRISPR activation is that like it is very, very strict in terms of like the distance dependency. So if you can see right here, when we're trying to put the CRISPR up front of the promoter to activate the gene, we can see that like if we move one base at a time, you can see that like the activity go up and down periodically. So at a certain part, it can has a very high activation, and it will drop if we move only a few base further. But it will come back at 10 to 11 base after we keep moving. So what's going on? Why is it behaving like that? The reason behind that is that like these 10 to 11 base periodicity is pretty similar to how the DNA fall. So what we might be seeing is maybe like when our CRISPR complex is at the right position, it was functional properly, but if it moved maybe like a few bases or like up to 5 base further away, it was on the wrong side of the DNA. So it may not work to recruit, to function as properly anymore, because like this part has to interact with some other thing in order to like make sure the gene is activating properly. So this is some sort of like restriction or like limitation that we've seen when we're doing our like, bacterial CRISPR activation system. We don't really see a lot of these limitations in the eukaryotic system, like in yeast and mammalian cells, mainly probably because like the original biological like system that was used for the gene activation is practically pretty different. Okay, let's move a little bit faster and like see what can we do further. Let's say if we want to engineer the RNA, we can also like we did try a few RNA to see like is there any effect that was dependent on the RNA, and you can see that like if we use different RNA RNA that we can have like different functionality or property as well. So the reason behind this is that we believe like for a certain RNA they can form or fall into a different structure, and what we are trying to tackle here is we're trying to develop that computational model, like predict first if the RNA is going to work properly. And if the RNA seems to be working properly in the computational models, then maybe like we can increase the chance of like the new RNA to be designed to be like highly functional, like this blue bar. Because sometimes if we like try to do like multiple things together, and like some of them were like this blue one, and some of them were like this purple one, then it's going to be harder for us to evaluate how these tools are functioning. The last thing I want to mention is like we can also like use the engineered Cas protein in order to expand what we can do in biology as well. So for example, if we are trying to like target any gene in the bacterial genome, that we might see like some limitations, because like we have to go to a very specific site in order to do the activation, right? And that site, if we were to target it, we will need the PAM right there as well, because like when CRISPR is trying to target something, it requires not just the complementary RNA, but also the part of PAM that has to be recognized by the protein as well. So in this case, we screen or try or tested like several engineered proteins that can recognize different PAMs. And what we found, like computationally, is that like if we change the protein from the original native dCas9 into the PAM expanded variants, we can increase the chance of like targeting the gene in the genome from like maybe just half of them into like almost all of them. So from this bar right here. And this is like the result that we have, just a snapshot from one of the paper that we published, that like if you change from dCas9 and you target something that has like these NGH and SG or like some different PAMs that is not the native PAM, it will not be functional in the original like Cas protein. But if you use the engineered version, it can has a much higher efficiencies. Like if you take a look at the B panel, you can see that like the blue and purple dots are usually like much higher compared to the original gray dots. And similarly on the right hand side on the panel C, you can see that these two green dots from the engineered Cas9 variants, they sort of like have a lot higher property in, or like the full activation, which is the number that we get from these like CRISPR screening to be like, you know, performing much higher. So we like, long story short, we can engineer at the dCas9 level as well in order to make the functionality of this CRISPR system better. And it's not just like the CRISPR activation system. This can be applied to the other idea of like the CRISPR gene editing or like the CRISPR technology as well. So finally, like what I want to mention is that we can also like try to port this system into the diverse system, to the diverse organisms. Like for example, if we have some bacteria in the industry that is not E. coli, not the model system that we want to work with, we can try to port it into the different organism as well. And the success like tools that we are using were predicted to be pretty promiscuous with other organisms. Like it can interact with different bacterial RNA polymerase alpha subunit or PLA right here. So we believe that like these two can be portable to a certain extent. And like, let's say if you have these CRISPR functionality tools, like, you know, happens in E. coli, like you can see that like if the guanate is not correct, they're not expressing this red fluorescent protein. But if there is the correct guanate, they can turn the cell into the red cell. So that's sort of like what we have in E. coli originally. Can we do that? That's similar stuff in another organism, let's say Putida. So P. putida is like another organism that was like pretty famous in the past few years because it's something that was like very tolerant to the harsh conditions or like, you know, some extreme chemical conditions. It can also eat a lot of like diverse range of chemicals. Let's say you would take some grass, some greens that you know, that can be chilled down a little bit. Most of those small molecules or like chemicals that were extracted from those greens can be fed into Putida. So that's why we want to develop these tools in Putida. And if you were to put this gene in, then we have to think about how to put it in appropriately. And what we found is that like we find that like if we put the big genes like, you know, the Cas9 and the activated gene to the genome, we can find a much better system to make the CRISPR activation tools be active. So long story short is that we make it. We can port these tools to a different organism and they can still function and can do like different things. And not just being portable, but like the rules that we've learned from these different systems can also be consistent. Let's say we do this kind of plot by comparing Putida and E. coli together, we can see that they're relatively consistent between the two systems and the rules remain the same from what I've been talking earlier. And not just another organism, we also want to try to see like if we can see it perform in a different system, like, you know, the cell-free system, when we lyse the cells open and like try to use it instead of like using the living cells. This is another angle of synthetic biology that might be interesting to you as well, because like, you know, it's something completely different. Like instead of living cells, they can they can divide themselves and keep growing. We are using something that doesn't have any limitation on how to grow at all. So like if you were to work on something that may be deleterious or might be toxic to the cells, you can use the cell-free system to understand how biology can react with this system that, you know, that you cannot do in the living cells because it's gonna die. And similarly, like this plot right here shows some consistency as well. And we were using this system to develop some new circuit based on the CRISPR tools that we have. And finally, we can use these tools to design at the gyne level to tune the expression level. You can see right here that we have a few ladder of like CRISPR activity provided here, and we can use it to tune like different genes and get different like chemical production profile based on you know, like tuning of 3 different genes. So that's something that we just recently pushed out, and you know, after that we can sort of like use these tools all together and combine with different things to come up with the new ideas of how to make a new system or a new production platform based on the what we call DBTL cycle, or like design, build, test, learn cycle, which is like one of the important ideas in engineering biology and synthetic biology, so that we keep design new things, do some tests, and then learn what we have done better, and then like try to design something even better. And most of the time, this DBTL cycle can be like pretty like difficult to do, or like takes a lot of effort. So like if we have something that is easy in both like the build, taste, and learn to come together and work, do a better job, that might be you know, something that could be useful in the future. So I think that's it. That's it might be kind of quick, but like we can talk further offline if you're interested in any of these things. Because like this is still a beginner course, so I want to give sort of like, you know, an overview. So like no worries at all if you don't understand completely what the specific part or like the specific like function of these things should be. It is complicated, and even like the PhD researcher that were on the team still, you know, take some time. They have to take some time in order to understand what the different parties gonna do. But if you're interested in the you know, the work, you can check these papers that I sort of like mentioned here. And I would say let's take that as you know, the end of our beginner course in terms of lecture. We can still talk for the time that we have left, and I will the quick question that is available in the chat, and maybe that's it. Okay, just stop sharing first. So the question that I see in the chat that maybe we haven't get there yet is CRISPR technology definitely is a game changer, but we can get hands-on training. I think that depends on the you know, how the organization is like scheming. I would say CRISPR has been pretty easy to implement these days. So it's more about like whether we can do molecular biology or not, or like rather than like whether we can do CRISPR or not. So like, you know, Heidi like explain that in the chat, and I hope like that sounds good to you all. And if we get to the stage that we can do some hands-on training, I believe like we will be interested in like putting some like CRISPR related stuff into there as well. Is there any specific gene or pathway affected when Cas protein is deactivated? I would frame it into a different angle. So like if you put the Cas9 protein or like some of these new genes in, there might be something that was affected. So for example, you put a lot of Cas9 into the cells, then at some point they may just like be that we may accumulate too much of them, and it may affect like the cell's growth to a certain extent or something like that. So I would you know, like narrate it in a way that like having new things into the system can affect some other things, but we can gauge in terms of numbers how much is affecting the other things. So if the change is very small, like it's changing like, you know, less than 0.1%, maybe that's not something we worry too much. But it's changing, let's say like 10% of the genes, for example, that's something that we have to worry a lot. So it depends. It depends on like how much expression level and how and what we are trying to express. Is there any finding or support the understanding of Cas protein deactivation? Yes. So most of the work that I was talking in the second part of the gene regulation is based on Cas9, as Heidi mentioned. and these were deactivated or nucleus date or something like that. D can stand for several things, but like mainly it can bind DNA but cannot cut anymore. So there's a lot of research that was based on these ideas.
There's a question I didn't get to that's nice from Dan. How does one measure the precision of CRISPR-Cas9? And what are the ways to reduce the potential off-target effects of CRISPR-Cas9? I mean, I'll just say this is like the $1 million question that defines whether or not this is safe to use in humans, which is, and maybe also agriculture, like anything that we're just gonna like let loose in the environment.
Yeah, so like that question, I would say, yes, we are aware of that, and we can do use some other technology like the sequencing technology to verify is there any other edits apart from where we want to edit as well, something like that. So we have some other, like, you know, there might be some specific tools like like OpSeq, something like that, you know, that something was specific for like trying to figure out what is the off target, because it's something that we care a lot. When we're trying to fix one disease, we don't want to generate another disease, something like that. But my answer on this, like how can we reduce it, I would say, like, take a step back onto how CRISPR works. It's based on 3 different things, right? It's based on the protein, the RNA, and also the target DNA. So in that angle, we can try to optimize each part individually. If we have like a better Cas protein that is working much better and it doesn't have a lot of off target, if we can design a guidance that would be much more specific, or we can screen the DNA of like which one would be the appropriate site for us to target, something like that, then we can have much better like fidelity and efficiency of the CRISPR system. And now has a question about like trying to use these in plant pathology or mycology. Yeah, there's a lot of like knowledge in the field. I see that Heidi has, like, you know, provide some information about that. So if you're interested, I would say like one organization that both Heidi and I are part of is EBRC, Engineering Biology Research Consortium, and we have some discussion on, like, you know, what is the frontier science and how do we use these tools to like tackle like the global problems. So I would say if you visit the website and see like what is we have the system called like road mapping, like we are trying to figure out like what is the bottleneck of the field and how can we use this to solve like new problems, and you may find something interesting to the new field or the field that you're working in around these documents that we keep posting. Okay. Maybe I see the last question from maybe like Water. Water said, like, can we use this technology in cancer therapy? Yes. But again, it has the same limitation. We have to make sure that we cure cancer and not making another cancer. So like we have to be very confident that there's no off target. And to do cancer therapy, there are like, you know, like billions questions out, billions like investment out there on how to treat different type of cancers. So it's going to be relatively hard to pinpoint what is the most effective way, but there are multiple ways that people are studying and like trying to optimize so that we can cure like different type of cancer or tumor in different kind of tumors or cancers, I would say. Cool. Okay, so I think that's all the questions that we walked through. Thanks everyone for asking a lot of questions and like you know, expressing interest in this research. If there is any other question, please feel free to drop it into our Discord channel, or that you feel free to email us as well. I'll provide these slides into our into our email later, and also in the Discord, so that you can check it again. And maybe this is the time for us to like check maybe other housekeeping thing, Heidi.
Yeah. Thanks, Ice. That was really awesome and so thorough. I think, yeah, again, there's a lot of things going on. I hope what you kind of take away is like CRISPR is a pretty general technology. I mean, asking what you can do with CRISPR is almost like asking what can I do with cloning. It's like there's so many types of plasmids you can build, and I think scientists are still trying to even like comprehend what the set of things are. So even if you didn't catch all the particular details, maybe there's something in there where you're like, I have a problem that I think I could solve with biology, and then I could use CRISPR in some way. I'm like interested in learning more about this. I know you guys are all kind of at different levels. We have people in undergrad, masters, and at PhD level. So I think for each of you, what makes sense as a next step might look a little different. But okay, so in terms of next steps, there is our short-term future, which is the homework. So I've just put a homework link in the chat. And I just wanted to point out this homework is a little bit different. We have some required questions, so questions you must answer to submit, and they're all related to feedback to the course, because you guys have sort of been our own little experiment, but in education. So we are doing this for the first time, and we would love to get feedback on how to do it better. I think we're doing something really kind of strange, which is to run a course entirely online with people who have never met each other in person. And it's been really fun to connect with you guys across literally an ocean. But if there are things that we can do better, we would like to know that. So please answer the feedback questions. And then we have the usual scientific questions. And of course, you'll get credit for those things, sort of however many you answer. Then the sort of next thing in our future is finishing the course and getting sort of like the completion certificates. We're going to put together all of your grades and then email you. And I think there will be some kind of certificate that we send you. That should happen, I think, maybe a week and a half from now. So the homework is due in a week. Then we take 3 or 4 days to grade all of the submissions. We'll also check if there are missing homeworks, and then we'll get back with you. But that will be the end of the course. And that's really exciting. And I'm already impressed. I thought we were only going to have 10 people here, to be honest. So to see so many of you here today, it makes it really fun and meaningful for us. And it's like super cool to discuss these things together. So I hope you've enjoyed it. And then in terms of long-term future, so this would be like, what is SynBio4All Africa going to do in the future? We're going to offer an intermediate and advanced course. So the idea is we'll do sort of more topics. If you have ideas for what you want to do, that'd be great. But probably what we will do is we will use the same Google Groups link to send out an email advertising that course. And you can sign up at a Google form link like you did before, and then we'll have like a new Google group for the next thing. I think we're going to do everything on Discord though, so if you stay on Discord, you should also get notified. But we would love to see you in these future courses. And we're also thinking about how do we sort of maybe see what else we can offer. Again, in terms of resources, we're trying to do stuff kind of across the ocean. Everything is basically free. So we want to do kind of more useful stuff. But yeah, we're trying to think about ways that we can support you guys in terms of accessing synthetic biology and advancing your careers. Please share with your colleagues about the intermediate course. Like the more the merrier. It is like super fun to have this community. Yeah, so I think that's everything for me. So yeah, don't forget to turn in your homework. Don't forget to keep an eye out for your grade. And don't forget to keep an eye out for more classes we can take. And yeah, we really appreciate your feedback in terms of what you liked about the course and what we can do next. Does anybody have anything they would like to add or questions? We have about 3 minutes before we need to sign off, but feel free to unmute or put something in the chat.
So one thing I would like to add on that is like, thank you, Maureen, for like trying to share the information about this course. But I would say like not just the new course that is coming. We will try our best to make this course that we've done already to be available for others. Let's say if we want to take intermediate course, they have the chance to take a look at what's going on in the beginner course as well. So keep an eye on, and like when we have that available, we'll let you know. And please feel free to share that with your friends. And this could be a pretty good materials for them before like getting onto the intermediate course, for example, because like we would be appreciate some background that they know. Yeah, and look out for the recordings. We'll make that available as soon as possible. I've been getting better at like editing things on YouTube now. I feel like a YouTuber.
All right, well, it looks like we are wrapping up. So maybe, thank you guys so much for showing up and participating. It's been a real pleasure. We look forward to interacting in the future via your homeworks and emails, and hope to see you all in the intermediate course. I don't know what Bildad's comment means. If this, I mean, I think, yeah, things get easier and harder at the same time, right? Like once you master the beginner course, you're ready to struggle in the intermediate course. And then you master the intermediate course and you're ready to struggle in the advanced course. A friend of mine once said that, this is about running, that the miles don't get easier, they just get faster, which I feel like is a good analogy. Is like, I don't know if any of you guys do any sports, but your level of effort sometimes stays constant and you feel like you're struggling. But if you look at, say, for running your times, you see that you get faster. And I think that that's hopefully the way the course should feel like, because learning is always hard. But if you look at what you're working with and what you've done it before, you will see that you're improving. And again, that's something else we kind of want to build, is like an exercise regime for your brain, right? Like what's the most effective way to like learn and get better? That's why we include the homeworks. Yeah, but you guys are all, I think, will be excellent runners.
Yeah, we usually compare like doing research as doing a marathon, right? So I'm pretty sure like you feel that, like, you know, like after completing the first course, the second course will be relatively easier, and you know, it will be more challenging and way more fun going on into like the deeper science, something like that.
Cool. All right, I actually have another meeting right now, so I'm going to head out. Thank you for, really appreciate all these kind comments in the chat. It really means a lot. Yeah, but I'll sign off now. If you guys have any other questions, feel free to put them on Discord. But I'm gonna, I think, end the meeting now. But yeah,
Thanks a lot,
good to see you
Heidi.
guys. Bye.